Well, hello everyone, and welcome to Race Oncology's Investor Briefing. My name is Jane Lowe, and I'll be your moderator for tonight. And it's also my privilege to support Race with Investor Relations. Joining me tonight are Race's Executive Chair, Dr. Pete Smith. Good evening, Pete.
Hi, Jane.
Hello. And Race's CEO and Managing Director, Dr. Daniel Tillett. Hello to you, Daniel.
Hello, Jane. Hello, everyone.
Hello. Okay, so now we were here a few weeks back talking about Race's science. Tonight, we're going back into the same zone. The good thing, though, once again, is that we'll also talk about the commercial benefits of having recently confirmed the anti-cancer mechanism of action for Bisantrene. The format we'll follow will be Pete and Daniel will talk us through a presentation, which was released this afternoon to ASX. After that, we will open up the floor to Q&A. If you'd like to ask a question, I can see there's already something queue. You can do that using the Q&A ribbon at the bottom of your screen. But for now, Pete, I'll pass across, or Daniel, pass across to you to get us on the road. Thank you.
Thank you, Jane. I'll just bring up the slide deck here. So welcome, everyone. Everyone knows we love science at Race, and so expect a fairly intense science seminar, but hopefully we'll give you a bit of a background into how this discovery came about and I guess, kind of the human element that goes into all of this. None of it's easy, but it's very rewarding. So I won't spend much time on this slide, and I'll give you this is sort of the one-page takeaway, as much as we can compact it down to being, how does Bisantrene work? And really it all revolves around these structures, folded structures called G-quadruplexes, and I'll talk a bit more detail about them as they're coming up. And they exist in our DNA and RNA, and they're involved in regulation of many important genes. They effectively act as switches, and they can turn genes on and off, and they cause and allow cancer to grow. They're also really important for normal cells as well, but when they go dysregulation of gene switches is a major driver of cancer. So what happens is, and I'll show some more detail as we get into this, Bisantrene binds to these G4 structures. They're dynamic inside the cell. They form and disappear continuously, and they stabilize the DNA or the RNA into the G4 structure. And this has a whole lot of effects, particularly around cancer genes. So there's a whole range of cancer genes that are driven by these G4 structures. And the one that we're really interested in is one called MYC. And Pete will talk about this in a great deal more depth than I will. And it's really important because it's involved in so many different cancers and the regulation of the growth of different cancers. Also blocks the activity of other enzymes as well in the cell, such as telomerase, topoisomerase 2, very important for cell division and disentangling DNA in the process that occurs when you have to have cell division occurring. And finally, it also explains why we have the influence around M6A RNA, and that's really important as well. That's a regulatory system that controls translation, the conversion of RNA into protein inside the cell. And when M6A levels drop inside the cells typically become very aggressive and resistant to treatment. And so if you can increase the levels of M6A, you can make the cells less aggressive and less resistant to treatment. Ultimately, you can think of Bisantrene as more than just a traditional chemo drug that kills cancer cells. It interferes with the control systems of the cancer. And this is really important because it makes the cancer more sensitive to a whole range of other drugs. And knowing this piece of information really adds a lot of value in deciding on what combinations of drugs to use in the clinic and which ones are likely to work, which ones are not likely to work. So before we get into all of that, I'll just give you a brief overview of what a G-quadruplex structure looks like. They're basically 4 Gs in a row in a DNA sequence, and they can fold up. Typically, they have a metal ion in the middle of them that stabilizes them. But the G nucleotides, the base, stacks it basically on top of one another and creates kind of a little crown-shaped structure. And they can form all sorts of different structures depending on the location, the sequence. There's just an enormous amount of diversity. There's more than 80 different types known They all have different effects. They're all bound by different drugs. So as you can imagine, the complexity of this gets really important. But they're really important at regulation of cell functions. Telomeres, which are the structures in the end of the DNA that allow DNA replication to occur, transcription, the stability of the genome. If you don't have a stable genome, you're going to end up with cancer. DNA breaks, translation of RNA, and also epigenetic modification of both the RNA and DNA level. So these very important regulatory systems that exist throughout the genome. So targeting these will have a lot of effects. And depending on the particular drug that binds to them, you will have different effects. And that's one of the reasons why different G4 binders behave differently to one another. They don't all bind the same way to the same G-quadruplex structures. So how did we make this discovery? I can't say it was easy. It took a long time. And the reason why it took so long is we were stuck with, the more we investigated Bisantrene, plus looked at the literature from the past, is the more things that we found. And it really was like the blind people, looking at an elephant. Depending on which you looked at, it looked completely different. Does all these different things, inhibits DNA replication and RNA transcription, inhibits angiogenesis, which is forming of new blood cells, which is really important for cancer, inhibits telomerase and cell division, inhibits TOP2, leading to double-strand breaks. That has been known for a long time, very important. Had all sorts of effects on stimulation of the immune system, very interesting, particularly around macrophages, which are a sort of a leader in the immune process. So they actually come into we have a tumor, and they'll come in and they'll see that this tumor is foreign, and they will then bring in the soldiers, the T cells that will come in and kill the cancer cell. So they play an important role there. They also modulate the M6A RNA epigenetic pathway, which we've spoken about many times in the past. Importantly, it's very important in the regulation of MYC. So we saw that MYC came down when we added Bisantrene. This was seen by other groups as well. It's not just us. But how do we explain? And then we saw a lot of synergy with other anti-cancer drugs with no real rhyme or reason to them, particularly kinase inhibitors and methylase inhibitors. And all of those go together to go, well, gee, how do we explain all of this? And there's really two ways you can go about it. First solution is the easy solution, which is we just ignore it all. We just go, oh, Bisantrene is this wonderful drug that does all these different things, and who cares how it does it? It's just a traditional chemotherapeutic like doxorubicin, and we'll just continue on that way. And that was the strategy that had been taken for many years at Race. That was one that I was never comfortable with. And the other alternative is to actually spend some money and do some real science and actually try and work out how all these things come about. And they're not just random. So the first clue we got, this is from about 5 years ago, actually. This is one of the earliest pieces of data that really, we went in and we did a cluster analysis across, this is an unbiased look at across 102 different cancer cell lines, across a wide range of different cancers, with 195 different drugs of all different mechanisms of action. And you look at that and see how do the drugs cluster together. And if you look up the top there, you can see all the anthracyclines, epirubicin, doxorubicin, daunorubicin, and mitoxantrone all cluster together. They all behave almost identically to one another. So all the cells that will respond or be sensitive to any of those drugs all work together. Bisantrene sat out with a whole range of completely unrelated drugs. And we go, what is it doing? It's not like anything else, which was kind of known in the past. It wasn't like the doxorubicins, really, or epirubicins, any of the anthracyclines, or even like mitoxantrone, which was the other drug developed by Ledley. It was different, and it didn't have the cardiotoxicity. There were many cells that were resistant to doxorubicin that were sensitive to Bisantrene and vice versa. It didn't behave in the same way. But one of the things that we did notice is one of the drugs there, tinomycin D, is a G4 DNA binder. And that's kind of one of the first clues that we were onto something. I have to say, we only noticed this after a long time. Second clue, which we knew pretty much from 2020 when it was published in the original City of Hope paper, where they saw a very strong decline in MYC. This key regulator takes a very small amount of bisantrene, or as they called it, CD1, no, CS1, I should say, not CD1, CS1, and that was in AML cells. We've also gone on and done this is in breast cancer. Different cells behave differently. They have different levels of expression of MYC, and they have all sorts of other factors that go into that. So they're not all going to be sensitive at the same level. But they also saw a really strong inhibition of MYC. And this is an interesting question because MYC is such a key drug, and people talk a lot more about this. And it's been a real target for many, many years for the pharmaceutical industry without much success. And then the third clue, this was the last sort of clue, we've been really struggling to try and explain how does bisantrene regulate the M6A system. We knew that it increases the level of M6A in RNA. We have shown this consistently ourselves internally. Lots of other groups have shown the same thing. The hypothesis was from the City of Hope that it was via FTO inhibition. We spent a lot of time on this area, and we were not convinced that we were on the right track. And we came across this paper, and really this explained how that a drug like a G-quadruplex binder could control the M6A levels. If you look at it from the perspective of it's a dynamic system, the M6A writers put on methyl groups onto the RNA, and the erasers, the FTO and alpha-PH5, take them off. And anything that changes the balance of those two systems will cause either an increase or a decrease. So an increase in M6A levels can come about through two ways. You can either reduce the amount of activity of the erasers, or you can increase the activity of the writers. And it turns out that G4 quadruplex structures within RNA attract the binding of metal 14, which is part of the complex that does the writing, and increases the activity of those. So what happens is stabilize G4 quadruplex, you increase M6A methylation, and that looks the same from outside as inhibiting. It basically mimics the effect of FTO inhibition. Very interesting discussion. And at this point, it was kind of like a small stone at the top of a hill, and an avalanche of discoveries were then made after that, where particularly Sumit, who's one of the new scientists that came in last year, joined us, identified mechanism after mechanism of the things that bisantrene does that are regulated through G4 complex or binding to G4 complexes. And that initiated a series of internal projects to look to make sure that this was actually occurring, which it does, and then we were confident that we were on the right track of explaining. And all the mechanisms of action that bisantrene has inside the cell can be explained through binding to G4 quadruplex structures, either in the DNA or in the RNA. And that really unifies the whole mechanism of action. It leads to a lot of downstream effects and makes bisantrene a really interesting and powerful drug, but we have one primary mechanism of action driving all of this activity. This is the eureka moment. This is all the various activities that are involved, and it really does provide a lot of value for us. It allows us to identify which are the best cancers for this to work in. Obviously, the obvious one would be cancers overexpressing MYC are going to be a really interesting target to go after. But across the board, it gets a good idea of exactly which cancers to go after. Even from my perspective, what's even probably potentially even more important is what other drugs to combine with RC220 in the clinic. There are hundreds and hundreds of drugs. If you combine that with the hundreds and hundreds of different types of cancers and subtypes, very quickly you get into an exponentially large problem of trying to identify what are the best opportunities out there. And knowing the mechanism of action allows you to rationally choose those. They won't always work. And it's certainly something you'll always find when you'll take two drugs that should work really well together and they don't. But the probability of success is just vastly higher if you know what you're doing. The other thing that's really important is it allows biomarker development. If you don't know how your drug works, you can't make a biomarker for that because you don't know what to actually look for. And drugs with biomarkers, tests that go along with that to identify the patients that respond or won't respond, have a much higher chance of success, more than double. One literature I saw was between 7 and 14 times more likely to succeed, which is quite remarkable. So this biomarker not only, and it also reduces the cost down, because instead of having to run very, very large trials looking for small effect sizes, you can run much more tightly where you look at only the patients that will respond to the drug, and you see very large effect sizes. So all in all, it's an amazing breakthrough for investors, because it just allows us to do so much more quickly with so much less money. And I'll just show you a little video here. This is created by another one of our scientists, Rodney Cusack, one of our principal scientists. And this is a model that he developed here of, you can see there at the top is the sort of, I guess, skin-colored structure at the top. You can see the 3 rings, and that's bisantrene bound to the top of a G-quadruplex structure here, stabilizing and keeping that structure together rather than wanting it to unwind and unfold. So it's a very interesting, scientifically interesting area. It's an amazing opportunity to go after. It's an area where there's a lot of interest in G-quadruplex binders because of the effects, but it also opens up a huge opportunity in MYC. And I will hand it over to Pete, who is the expert at race on MYC.
Yeah, thanks, Daniel. Yeah, so just a little bit on that. So I did my PhD many moons ago in cell signaling, and that's basically exactly the area that we're talking about now. And I stepped off the board of a company called MYC in January, having been on the board for 7 years. So spent quite a bit of time looking at, thinking about, and getting to know MYC. So it's sort of really amusing that we've ended up where we have, and I did bore a lot of people in race over a very extended period of time, saying, "I think this could be MYC that's having this effect." So anyway, but it takes a mountain of people to do this, and I've said it before, and I'll say it again. The race team rather is so engaged with what we're doing, and as Daniel said, a lot of people have been involved in this process of working it out. And I'll talk a bit about MYC. Don't worry, I won't go on for days, even though I could. And it really is. It was one of the very first oncogenes to be discovered. So it's actually the viral. It was discovered in a virus first, an avian virus that infected birds. It's the myelocytomatosis virus. And what happened back in the early days of discovering oncogenes was they were quite often found in viruses that caused cancer, and then they'd find what are referred to as homologs, human homologs. So they'd have a very similar sequence, and they'd work out how they worked. So I'll touch on KRAS. That came from the Kirsten rat sarcoma virus, and there was SARC and VSIS. And just one funny story, actually, when it was discovered that VSIS was a human protein or coded for something very similar to platelet-derived growth factor, there was a newspaper headline in The Sun, which is not renowned for its science coverage, and it said, "Cancer jab on way." So that was just a quite amusing thing. Of course, it hasn't exactly turned out that way. So MYC itself is considered to be undruggable. And part of the reason for that is, and you see these helices, these are relatively unstructured domains, and it's really only when it binds with a partner, and its main partner in terms of activating it is a protein called MAX, and then that can then bind to the DNA, to sections of DNA called E-boxes, and that then will switch on a very, very broad range of genes. So depending on the amount, so it is dose dependent. There are high affinity sites and lower affinity sites. So the high affinity sites, you may have 2,000 genes that are being switched on by MYC. And when you've got a higher level, it will switch on lower through lower affinity sites. So there's a sort of a dosing effect there. So it's a transcription factor, one of many transcription factors in the body, but it is the master regulator of basically growth activity. So be that development of the embryo, the development of tissues, wound healing, angiogenesis, MYC is involved. And so it is a really central regulator. It affects more genes than any other transcription factor. And it is, as Daniel mentioned, it's upregulated in 70% of cancers. The way to think of it is, if cancer is a fire, MYC is like a can of petrol, and it will accelerate any cancer where it is upregulated. We'll talk a little bit about how it is regulated itself. So it is this potent accelerator. There aren't any direct inhibitors in the clinic at the moment. There is a peptide called OmoMIC that's in development, but I think that has challenges in terms of bioavailability and activity. So this is this chap here, we can call him Mick, I guess. MYC really is this holy grail target. People have been working on it. As I said, it was discovered in the early 80s. People have been working on this ever since, trying to find drugs. You can find inhibitors of it, but they tend to be very, very low potency, stuck in the micromolar range. And as I said, 70% of all cancers have increased levels of MYC. And that means that there's well over 1 million new cases of cancer in the US alone that have elevated MYC. And as we come on to in a later slide, MYC is involved in basically all of the hallmarks of cancer. So you can actually tie back nearly all of the things you associate with cancer to the presence of MYC. And believe me, I know this all too well. There are 53,000 papers written on MYC, the vast majority of them in cancer. So, several thousand a year. It does take quite a bit of effort just to stay on top of the literature. The next slide. Yeah. So this is why it's important. So if you look on the diagram on the right, you've got a number of pathways from growth factors, survival factors, cytokines, inflammatory cytokines like IL-6, developmental pathways like Wnt, and there are others. Actually, this is a simplified version, but as this very, very clearly shows, they all basically focus on MYC as a hub for their activity. And if you look in those pathways, these are the cell signaling pathways. I've worked on one that's actually not on this, but the hypothesis was that all the oncogenes would sit in these pathways. They're not all there, but take, for example, RAS is sitting there with a fairly central role in terms of growth factor signaling. It gets mutated and it can drive the cell. And in fact, there was a paper recently that said that if you have a cancer cell and you knock out mutant RAS, if it has mutant RAS, it has an effect, obviously, on slowing the growth down and changing the phenotype of the cell. But then if you knock MYC out, it looks exactly the same. So it's almost as if all of the signaling is being driven through MYC. That's not biologically strictly true, but it is one of the major factors. So MYC is upregulated by all these signaling pathways. And if in the literature, this isn't our data, but in the literature extensively, what happens is if you block, something like mutant RAS, or you block a tyrosine kinase, which is signaling a growth factor, MYC levels go down, and then resistance emerges because one of these other pathways has a mutation in it, and all of a sudden something else is to MYC overexpression or overactivity. So I won't go through all of these here. The effects on the stability of MYC, that 15 to 25%, that's things like M6A. So when M6A is high, it leads to the destruction of the signal, the mRNA that's coding for MYC. And so that's why there is this, it looks like an FTO effect. So interestingly, mutations in MYC itself are actually quite rare. And I won't go into the sort of thinking around the biology there. But again, I think the important point is that MYC is this hub of all these signaling pathways that are known to be driving cancer. Next slide. And so these are what are referred to as the hallmarks of cancer. On the left-hand side, we have all the useful things that MYC does in a normal cell. Wound repair, stem cell differentiation, etc. You can read that. But on the right, you've got these hallmarks of cancer. Uncontrolled proliferation makes perfect sense. Immune escape. The tumor microenvironment is very hostile to T cells and other immune cells. And that is very much a process driven by MYC. MYC leads to immune checkpoints being expressed. So things like PD-L1 are expressed on those cancer cells, and that's obviously what Keytruda and other significant checkpoint inhibitors work on. Metastasis, it drives metastasis, it immortalizes the cell. And again, that there are multiple ways that can happen through telomerase and others. Angiogenesis, drug resistance we talked about, changes metabolism. So the cells switch to using basically just glucose and not bothering with all the hard stuff of taking the energy efficiently from glucose. It just runs through glucose, uses a lot of glucose, and that's why FDG PET, so that's a labeled deoxyglucose, that's why the tumors light up in those scans, is because of the high level of use of glucose. So very, very important. That's called the Warburg effect, if anybody wants to look that up. Anyway, so on to the last slide, which is the important one, really. And that is around knowing the mechanism of action of a drug. I can't tell you how important that is when it comes to talking to big pharma in particular. And when I joined, and again, I have said this publicly before, I said to the team, right, this is a boring old chemotherapeutic. We have got to find something interesting and exciting about it. My goodness, I think we've done it. So it really is important. And as I said, to be able to go in and say, right, we're having a specific effect on known structures, and therefore the effect is predictable, that is extremely important and valuable. And so, again, Daniel mentioned this. We can now go into those 53,000. In fact, the other day I looked, it was 53,333 papers on MYC. We can dig into that literature. Much of it we already know, but we can dig into that literature and look at different drug classes, have a look at the mechanisms of resistance. We can test those. We know where to look. And it's worth pointing out that nearly all of the work that's been done historically with Bisantrene, it was used as a monoagent. So there was no exploration of using it in combinations or no idea about looking for resistance within particular cancers. So, it really does help us. We can identify the patient populations to go into. We can stratify the patients to make sure we're treating those that have, high MYC or whatever other biomarkers we find. So it's going to be better for patients during the clinical trials, but also hopefully will really help us to rapidly focus in on the cancers where we're most likely to benefit. And again, biomarkers, all of that. And so this really is going to be a big push, a big positive push for our partnering. I think the one thing I would say is we need to understand what it is that we're partnering. We need to understand how broad this is. And I'll just leave you with the thought, I'll sign off after this, that had we done a deal, last year, the year before, for a modest upfront, we would have been leaving so much value on the table for shareholders. Going through this process, really working this out. This is what adds to the value to a program. So that's my half an hour up. So I'll stop there. So thank you.
Thank you. It's a great soundbite to end on too. Thanks to you as well, Daniel. Okay, so now let's open up for Q&A. We've already got about 20 questions literally in the queue. Let's start with this one. It's an obvious question. Does the MYC discovery have any impact or require changes to or delays to our current phase 1/2 trial in RC220? And Daniel, maybe just stops sharing your screen so we can see you.
Should I answer it? Yeah. Stop asking questions about future plans. I can't tell you. So, of course, everything affects everything. We can only announce price-sensitive pieces of information via the ASX. Once we announce it via the ASX, we will. However, think about this, knowing more about how the drug works, it can only help speed up that trial and make it more likely to succeed. So there may be modifications, there may not be modifications, but if they are, there will be things that speed things up rather than slow things down.
But I think overall, I think just to add to that, the fact that we now know more about how it's working isn't going to change the way it's working. It works. So in terms of efficacy. So in terms of the current trials, no, that's ongoing. It doesn't change, the cardioprotection effects that we see. It's not going to change the efficacy. What it will do is open up those new opportunities and those new avenues to explore. So
yeah,
right.
Everything always has an effect, but yeah, it's not a negative.
No.
Okay, very good. Excellent. Okay, Pete, this one might be for you. So based on the mechanistic discoveries, we would expect greater activity of bisantrene and MYC upregulated cells. Does bisantrene have a sensitizing effect on normal expressing MYC cancers? I know you've talked about that a bit, but
I think it will have an effect, and it will be cutting off the ability of that cell to then find a resistance mechanism as well. So, I suspect that any cancer cell we would see an effect, but it clearly will be having a greater effect on cells that are kind of got themselves addicted to MYC. So, there is a thing called MYC addiction. I thought I was suffering from it for a time. But,
sounds like it.
So I think it's still early days there.
Yeah, I would remind that the mechanism of action is G4 binding, not MYC. MYC is a downstream of the G4 binding,
Yep.
and G4 binding has lots of other effects as well. So even in cells that where G4 is normal level, if some of the other pathways that G4 binding can affect, then it can have a direct effect that way.
Yeah, and we still have to explore, right, which of the 80 G-quadruplexes we bind to with high affinity, and which genes are controlled by those, really work out the pattern of activity.
Yeah, I think it really needs to be emphasized. This is a recent discovery. It's only a little over 6 months ago that we developed this hypothesis. We started testing it, yep, we're on the right track. But there's so much to explore. This is just an amazing opportunity for doing great science. Now you've got a great drug. So it's really early days there's so many things that, so many experiments that I want to see done. It gets ridiculous. So yeah, and we will. It means there will be lots of news flow to think about if you're an investor perspective.
We like that. Okay, so nice segue here. Will bisantrene enhance Keytruda, where the cancer quickly proves resistant to Keytruda? Pete?
It's definitely something we're going to be looking at. As I said, MYC is absolutely associated with the hostile tumor microenvironment. We know about the macrophage polarization. That's known. So I think whether something like Keytruda, whether it's going to work alongside Keytruda, I think it's something that is screaming out to be tried. More challenging than you'd think, because you have to go into an immune competent mouse to look at that initially. You can put human immune cells into a mouse lacking an immune system, but it's challenging to do that. But it's screaming to be looked at. Absolutely.
Thank you. Okay, Daniel, this one might be for you. Similar vein. Does race have any human validation of this G4 in brackets MYC MOA occurring?
Well, it's kind of an interesting question, because there's lots of human validation that bisantrene works, or bisantrene works already in humans. It's been in 1,500 patients. It's been approved, so we know it works. What we didn't know is how it worked. So really what this is a back validation. So you got a drug that initially was thought to just be a chemotherapeutic agent. Now we've worked out how it actually works. So that type of question is different to you normally have with a new drug. So normally with a new drug, you start out with a target that you go after, and then you try and work out whether that target's important in cancer or not, whether it'll work. And most of the time it doesn't. Like maybe one time in 100, it'll actually work out. We have the a huge advantage with starting with a drug we actually know works, and then trying to figure out how it actually works. That's not an opportunity that many companies have. I much prefer that method because the probability of success is much higher, because once you figure out how it works, that's it. You now know, and then you also know that the drug works in humans. So you've done everything you need.
Right.
I'll just add to that as well. One of the things that people have been concerned about MYC inhibitors is the toxicity profile, because it has this really fundamental central role in so much of biology. You know, is taking MYC activity out altogether, is that going to be extremely toxic? And we've got the answer to that. No, because we've got the historic data on Bisantrene and all of those clinical studies. Personally, I've never been concerned about that. There are, you can't knock the MYC gene out completely in mice because it's so important for development. So the mice don't grow. It's called embryonically lethal. But you can generate mice where you knock it out after the mice have been born. And so it's called a conditional knockout. You give them a drug and it, anyway, it gets rid of the gene. And those mice, they looked a bit knocked off and they looked like they were older. Their fur was all ruffly and they were grey, but they actually lived longer. So it's quite an interesting one. So maybe it's telling us we don't want to be profoundly inhibiting MYC throughout the life of an animal or a human. But actually the evidence is that it, if anything, it actually helps you to live longer. So it's there are definitely life, health span benefits. This is all in mice, by the way, no human data. But if you reduce the level of MYC by 50%, the mice live 15-20% longer, and they don't get a whole range of different age-related diseases. So again, it seems like it's an accelerator for the aging process as well.
And we've already talked about the potential anti-aging properties of RC220.
Yeah, and that's obviously, just to remind people, that's in the context of doxorubicin. So doxorubicin is progeric. It accelerates aging, and our hypothesis is, and again, this data supports it, is that we would be able to block that in the way that we can actually have a positive effect in the cardiotoxicity setting as well.
Very good. Okay. Pete, this one will be for you. I promise I'll come back to you, Daniel. Would you be able to provide a quick comparison of Bisantrene's MOA and efficacy compared to other indirect MYC inhibitors that have successfully completed phase 1 trials? Specifically, you mentioned this one before, OMO 103, PMR 116, and to a lesser extent, IDP 121, which is currently undergoing a phase 1 trial. Some homework done there.
Yeah. So I think based on what we know, we have a very active drug here, and we have all the background data on it. So we were getting excited today. We've got a phase 3 or potentially a phase 3 MYC inhibitor. Certainly OMO-MYC, that has been around for a very long time, and as I said, it has issues in terms of delivery. And I think if you look at all of the other drugs that are out there, none of them is particularly potent. They all have deficiencies, and it's because rather than most drugs bind to a defined pocket in a protein, which is like it's like a solid lump. It's got a tertiary structure. It's 3-dimensional, whereas MYC has just got these. This part of it is just basically blowing about in the breeze, so it doesn't really have a fixed structure, and that's why it's been so difficult to pin it down. Because Mike would be able to give chapter and verse on this, but what you need is a starting point, and then you have you develop a structural activity relationship. So you need a negative charge there and a positive charge there and a hydrophobic group over there, and then you play with that and try and make it more effective. But you've got a starting point and you can rationally build it out. It's just been very, very hard to get structures and to really understand how to build a molecule for MYC.
Right. That was Professor Mike Kelso that we were talking about, who was on our last webinar. Not the guy on the slide back there.
Maybe he could be MYC.
Mike, not MYC.
Exactly. Okay, Daniel, over to you. few questions here on biomarker development. We might start with, could you tell us the progress of biomarker development? Then we'll add to that.
That's in general, like, I don't know
Yeah.
if really much more I can add to the biomarker development. We won't be making any announcement about any race biomarkers, except via the ASX and post any patenting. So we can't really talk about anything that we're doing internally at this point, unless the shareholders want us to destroy the value of their company in the process. I assume they don't. So yeah, but knowing the mechanism of action makes developing biomarkers so much easier. So you know what to go after.
Okay.
And you can be assured that we're fully aware of the importance of biomarkers. We're now aware of the mechanism of action, and assume that we're not a bunch of idiots that we will be adding 1 + 1 and coming up with 2.
Okay. Apologies to the other people with biomarker questions. I'm going to mark those as done based on that response. Got one here. Excellent work, team. Pete, if
Well,
you're
no,
working on
no.
the RCR.
I mean, I think it's a G-quadruplex binder. That's how we need to refer to it. And the reason we're talking about MYC is that so many of the effects in cancer can be ascribed to reducing MYC activity. We, as I said, we need to really explore this. There are techniques out there. There's some excellent groups, and as we find out more, we'll be able to ascribe more activity to maybe other proteins that are downregulated. It's very complicated because, as I said, MYC is a central transcription factor. It is turned on by transcription factors, and it also turns on transcription factors. So you actually have this sort of cascading effect of, a bit like a snooker ball hitting whatever the triangle thing is. And balls going everywhere, but it's the white ball hits only one, but it can have an effect. And working out what's primary and secondary. But again, there's been so much work done on this. So when you look at data coming out from your cells, looking at what genes are being expressed, there are classic mixing signatures that come up. So it's called gene set enrichment analysis. And so you have MYC targets 1, MYC targets 2, and people will be seeing data like that coming from us as we determine this. So those will be like the first row of genes that are getting activated, but then they will go on and have effects as well. And it is, as we described with the M6A, FTO, it's labyrinthine in complexity, and nothing is happening in a straight line. It's not A goes to B goes to C. It's quite a chaotic process with feed forwards, feedback loops, proteins being tagged for destruction, mRNA being tagged for destruction. It's there's a lot going on, and to try and think of it as being a linear sequence of events is sometimes not the way to go.
Yeah, but it is all coming about through G4 quadruplex binding. So that's the cause. That's what it is. The G4 quadruplex binder. There are a few out there already. Some of them, they've got problems, issues. It's very difficult actually to make a G4 quadruplex binder, because the properties that make it a good binder to these G4 structures make that a very poor drug. And in fact, Bisantrene is a terrible drug from a drug property perspective. Ledley had enormous problems actually trying to get this drug into patients and not have it immediately crystallize in their veins. We had the same problem. Ledley gave up. We persevered with that and came up with the RC220 formulation. And we wouldn't have done that if it wasn't already developed as a drug. If it would have been invented in the lab, a couple of years ago by one of our scientists here at Race, we would have immediately thrown it out, would never have even made it into a mouse, because the properties of it are so undrug-like that it wouldn't, you wouldn't even think of putting it into a mouse, let alone a human. And so those unusual properties are what make Bisantrene such an unusual drug, and then work in such a different way to all the other drugs. So G4 quadruplex binders have had real problems, the chemists have had real problems making them into drugs, and vice versa. There's just not many around. So it's a really I guess we're just unbelievably, I'd say, lucky to be in this space where it's a really great target to go after, has all these amazing effects, particularly on MYC, but also on other systems. And people just can't make these drugs, and we happen to have one. So and it wouldn't have come about if it wasn't for Ledley, wasn't for them struggling away with it. They persevered with it. They saw something special in it, despite it being a real pain in the neck to work with. And we build upon all of that. Luckily, they didn't notice that it formed isomers, so that's something that we could discover later on, which obviously forms the backbone of our
And I
IP
think,
now.
and I think, I mean, I actually posed the question: well, why hasn't Bisantrene, Bisantrene, or whatever form of it, why hasn't it turned up in random screens? Because people would have been doing high throughput screening for things like G-quadruplex binders and things that affect MYC. And the simple fact of the matter is, you just wouldn't put Bisantrene into a screen. It could be for that reason. And you look at it; it's really not like a modern drug at all. It doesn't have three-dimensionality to it. It's boring, flat, planar structure with two little arms. And yet that's exactly what you want as a G-quadruplex binder. So I think it's almost by complete accident that we've ended up where we are with this drug. And as I said, it really is very undrug-like in that it's so insoluble at physiological pH. So you just screen it out for that.
Yeah. So what happens? You put it into a high throughput screen, and it immediately crashes out in that high throughput screen. And they're called pains, these drugs. They're drugs that just kill your assay straight away, and you just remove them from the thing because you cause endless problems to your assay. So when they crystallize out in the assay, it pulls down the protein in which you're trying to target and causes all sorts of problems. So this is one of the reasons why you just don't see this sort of work. So it requires a really unusual series of combination of events to have occurred for these discoveries to be made, and really talented people to make those jumps as well. So it's not only you've got to have the right preparation there, but you could be sitting there. We could have just decided, oh, we don't really care how Bisantrene works. It's just another anthracycline. Who cares? We're just going to flip it off to somebody else for maybe $30 million, and we'll take off into the sunset. But actually deciding, that's not the right way to go about this. There's something special about Bisantrene. It's not a anthracycline. How is it working, and figuring that out really pays off with a huge amount of value generated for shareholders. And I think this really shows the power of preclinical research within a biotech company. Everyone's focused on clinical research and so forth, like this partnering. But if you don't have a good foundation built on preclinical work, then you're not going to get very far. So it's really the foundation, concrete slab that you build everything upon. You build it on quicksand, and it's not going to go very far.
Right. Well done to the team once again. It's terrific work. Okay, I've got my eye on the time. We've got 10 minutes to go, and we've got a lot of questions in the queue. So let's go rapid fire. So on slide 14, Pete, I think this was yours. Pancreas stands out at 97% having elevated MYC. Does that indicate this could be a very relevant future cancer trial area?
So pancreatic, was that?
Yeah.
Yeah. So we were talking about it earlier. Pancreatic cancer is where drugs go to die, but it's a huge problem. That is almost certainly to do with the fact that the vast majority of pancreatic cancer is driven by mutant RAS. So primarily, there are three main mutations in RAS: G12C, which is more associated with lung cancer, and then G12V, G12D, which are associated with pancreatic cancer. So that'll be why. So if you've got mutant RAS, it's going to be driving MYC hard.
Okay. Thank you. Okay.
The reason why pancreatic is so hard, it's very hard to get the drug into the cancer. So lots of drugs can kill pancreatic cancer cells. The problem is inside a patient, it's walled off behind all this fibrotic tissue. It's basically scar tissue surrounding the cancer cells, and none of the cancer drugs can actually get into the cells. And that's what makes it so difficult, cancer. That's why it's such a death sentence. And similar thing with brain cancers. So you have the same problem where the blood-brain barrier keeps out most of the drugs. And so it's very difficult to actually treat brain cancers for that reason.
Got it. Okay.
Generally, as a general rule, if you end up in pancreatic cancer, you made a mistake somewhere along the way.
Right.
That's your best option.
Or hopefully you've got an excellent discovery coming down the line, which would be good.
Yes.
Okay, so Daniel, for you, whilst this is a recent discovery, given the abundance of revelations that have come about and are yet to be shared, is there an expectation that this will open up opportunities outside of oncology? If so, can you share any thoughts on what direction that might look like?
Yeah, this is a difficult one. So Bisantrene has properties about it. It's that make it difficult to use outside of cancer. As much as it would work quite well, I think some of our shareholders remember back to the infamous diabetes, type 2 diabetes, and Bisantrene is very good at making islet cells in the pancreas excrete insulin, and turns around and makes islet cells that have become lazy and don't respond to glucose anymore, makes them active again. And that's a really amazing discovery. How that works, we actually don't know, but that was something that a group out of France discovered. But would you use something like Bisantrene as a type 2 diabetes drug? And the answer to that is almost certainly no. And the reason for that is the barrier safety level required for those types of drugs are incredibly high. It's very, very difficult to make a new diabetic drug because it has to be 100% safe in 100% of people 100% of the time. And that's very, very difficult to achieve. In cancer, the tolerability for side effects is much, much higher. Patients can be pushed all the way almost to death. As long as it cures the cancer, everyone's willing to live with that. And so it's a very different environment in which to develop a drug in. So, yeah, I would not like to be trying to develop a drug in diabetes. Not that there's not a great need, but the chance of success is very low because there's so many ways that your drug can go wrong. You can have one patient in 10,000 that gets liver failure and your drug is dead. If you have one patient in 10,000 that gets liver failure from a cancer drug, no one will ever worry about it. It's just part of the course. Bad for the patient, but yeah, not so much for the drug development process.
Okay, rapid fire. Moving on. Pete, is the FTO inhibition mechanism MOA upstream or downstream or totally independent of the MYC inhibition MOA? I think you covered this, but perhaps just close that one out.
As I said, I think we can ascribe the effects on M6A as being downstream of MYC, and obviously G-quadruplex depressing MYC and changing the M6A. We do understand that, and around the G-quadruplexes themselves. So I think the question is there room for FTO in this mechanism? Again, everything's going to be interlinked, and I think we see this all the time. So drugs like another G-quadruplex binder, pidnarulex, CX5461, has been ascribed several mechanisms of action before they realized it was a G-quadruplex binder. So biology can really lead you astray. And what happens is, a paper comes out and says, oh, this is how it works, as happened with the City of Hope with Rui Su's paper. And then everyone just says, oh, it inhibits FTO. And look, oh, we used it in this cell line. Inhibiting FTO does this. But they haven't actually gone back and independently verified that is exactly what's happening. And as I said, some things can look like others. You knock FTO out, it affects MYC, and so you have this effect that makes it, so the biology is complicated, and I think really homing in on, what's real. And I think the important thing is that the concentration of Bisantrene that results in the MYC inhibition is basically the concentration that works on cells. And that's very, very important. Sometimes people will say, oh, this is a, protein kinase B zeta inhibitor, but they're actually not using a dose that is relevant to the clinical setting. So it's almost certainly not what's happening in an animal or in man. And so, you've got to be very, very careful with the science and how it's interpreted. And as I said, people get led astray, and it's not bad science. It's just simply that it can be confused, and particularly in this area. So
cause and effect's always very hard to figure out. You've got dynamic systems and where there's multiple inputs into that system, can very easily go astray.
Yeah.
Yeah.
Okay. Daniel, a question here addressed to you. How many once in a lifetime opportunities can one drug have?
I don't know. I continue to be thinking, I was actually having a chat with Pete about this, about, Greek tragedies. Are we being set up to be cut down by the gods? Because unbelievable, like to have so many things go right. And it hasn't been easy. That's the other thing. It's not just been, it's all just fallen in our laps. It's been a real struggle. The mechanism of action has been something, I've been working on from the minute I arrived at Race. I wasn't satisfied that we just say this is a old chemotherapeutic and that's all we do. And it's been, the whole team's been, the same questions have gone around and around internally. How is this drug working? It has so many different effects. It's not just, it's not straightforward. How do we unify all of this? How do we find out what's causing and what's a downstream effect? And so that's been really amazing. And then, solving some of the other things like the IP situation, to be blessed with having a drug and being able to reset your IP, 40, almost, it's actually almost 50 years old. It was first synthesized in 1976 and patented in 1977, I think. Was the original, and patent got granted in 81. So that, you just don't have that luxury. And then to be able to restart again in 2025, with a 20-year life ahead of us on an amazing drug in an amazing area, that, really couldn't have been made before now. It wasn't because Ledley was, people working at Ledley weren't dumb. They just didn't have access to all the tools that we have today. So we've been able to take modern science, modern technology and figure these things out. And certainly talking to some of the old guys at Ledley, when we tell them about what we've been up to, that they're so amazed and interested in what's going on. So it's really nice from that perspective. So it's very satisfying for me, anyway, from a scientific perspective.
Yeah, that's great. Okay.
What's the likelihood of getting a MIC expert join the board?
I know, I was thinking that we might need to rebrand.
There's not that many people. There is a lot of papers and a lot of people who work on MIC, but there's not many people who've spent 7 years of their life before they join Race working on MIC. So I was aware of MIC. I wasn't, but to me it was just another one of the 1,000,001 cancer regulatory genes out there. And so I hadn't devoted 7 years of my life to MIC. So to have Pete come and join is truly
I was a non-executive director, so I think
the rest of the board.
I was a non-executive director, so I think devoting my life to it is a bit of an overstatement.
Come on.
But the cell signaling background as well is very helpful, Pete. Okay, hey, I've got my eye on the time. We're just about at 7 o'clock. We've got a lot of questions left in the queue. I think maybe we'll take a few more, and then if people need to jump off and have dinner, please do that. But so science is cool, Pete. But can you explain how much value this adds to potential deals or to the overall total addressable market?
I can't really answer that because, again, we need to define what it is we're selling. We have a number of drug classes that we want to look at in combination, resistance mechanisms, and really work out how best to deploy this. We are not going to be short of work anytime soon, and as I said, so I think there is, we need to make a investment in preclinical research, and then that will then lead to further developments, I think, clinically. But in terms of, as I said, I've got a spreadsheet together already with all the major companies, all the major anti-cancer classes that we think at this point makes sense to look at. Like, there's no point in us trying to see whether we can overcome oxaliplatin resistance. It's a generic drug. There's no one's going to get excited or pay us for that. Whereas some of the new targeted therapies, some of which have massive franchises, in the 5 to 10, or even higher $1 billion a year sales range, there are companies behind that would love to be able to keep them working longer, sell more of it, treat more patients with it. And so I think, that's where the real opportunities are going to lie. The other thing is that, particularly in the new targeted therapy space, it's very, very competitive between companies. And, increasingly we're seeing Chinese companies really stepping up with very, very, very good molecules, absolutely on a par or better in some cases than their Western counterparts. And so there's this real competition between the incumbents and the young pretenders. And if we can work with a young pretender, we can maybe turn their drug into a giant killer, or we can work with the incumbents and keep them in top spot. So we're going to have some very, very interesting conversations. But again, I think we can have those conversations now, but I think we really need to understand, right, does it work with that drug class? Does it work with this drug class? Does it work with the other? It is responsible for resistance to a lot of chemotherapies. So it's not just the targeted therapies. It's a very, very broad mechanism. But again, I'd be just waving my arms around if I said it's going to do this, it's going to do that. We need the science once again to lead us in the right direction.
Okay, so there are a couple of questions here about commenting generally on the characteristics of the right market and clinical trials required to access these discoveries. So what we're saying is we've got a good idea of where that might need to go, but we've got the work to do before we can answer that properly.
Yeah. And of course, that there is work ongoing. We have a number of experiments running right at the moment, which I think are going to provide insights. And I think, so the message is, we've had a lot of very significant news with the patents and now the mechanism of action, but I think everyone should expect that there will be additional information that we'll make public when we have the results. And mechanistically, you'd have to be very, very hopeful that some of these experiments are going to yield, given what we know out of those 53,000 papers on MYC. So,
yeah. So good question here for you, Daniel. Do these recent announcements on MOA help the interest in enrollments in trials? And I don't know if there's any comment on the current trials.
It's a huge one of the huge problems we've had historically with getting clinicians interested in Bisantrene or Bisantrene now is they go, it's an old boring chemo drug. I'm interested in targeted agents, new therapies. I'm interested in MYC, these types of things, and to be able to go out and explain. And we've already, the conversations we have, we're always talking to clinicians and sharing, the discoveries we've been making as we've been going along. And, the level of enthusiasm that we've seen in recent times compared to historical times is just off the charts. It's like I go to sleep one day and wake up working for a different company because the level of interest in what we're doing is just so much more greater today than it was 12 months ago. And I think and ultimately that translates, clinical interest translates into commercial interest, which translates into deals and dollars and all the rest of it. The problem is chemotherapy is, whilst it's there and being used in tens of millions of doses of chemo given every year, nobody's particularly interested in it, scientifically. Where they are really interested in G-quadruplex binders, they're really interested in MYC, and they're really interested in combinations that will have unique properties that can treat cancers that other drugs can't. And all of that goes together to make a really amazing opportunity that we now have, and we have to really build that up. It would have been great if I and the rest of the team had come up with this idea 5 years ago, although probably things would have turned out differently. So I think we need to be the right time, right people, right opportunity. And I think the last couple of years in particular has really changed the way race works, the way we go about things, and the discoveries we've made have all come from that dedication and hard work. The amount of hard work that goes into all this stuff is just, and get people to be willing to do that is impressive. And they're doing it because they can see the value in it. Like it's not just their options. It's
great team.
Science.
Yeah. Okay. Another one on sort of third party interest. So have the lines of interest and inquiry from third parties increased with these recent discoveries on MYC and MOA, noting this has all been fairly recently announced?
Yeah.
So
No, we haven't. You know, we one of the jobs ahead of us is to get the deck ahead, put together rather, with all of the new data in it. And you know, again, you got to time your run in business development. You know, we need to make sure that we've got sufficient high quality data in the data room and in the deck to convince a big pharma partner that we're onto something. And that is more challenging than you'd think. You'd think you'd be able to sit your head around the door and say, hey, we've got something that hits Mick, and everyone would be piling in, you know, wanting to do a deal. No, they don't work like that. They don't work like that at all. You need to, you know, really define that activity. You know, we need to, you know, do some genetic experiments, changing the Mick G-quadruplex, maybe removing it and showing that Bisantrene now doesn't work. Things like that, right? That's how you really define that mechanism as being real, that Mick really is part of the downstream effect of treating a cell with Bisantrene. So, you know, we need to get that done first. Now, again, we know what to do, because the literature guides us. And you know, we're working with some interesting people. And just a shout out to Daniel von Hoff, who has been a terrific advisor to the company and a great source of inspiration and direction. So I just mentioned him, you know, and again, the importance of those external collaborations and conversations. So yeah, sorry, I went a bit off topic there, but so
I will just mention briefly that there is a question here about whether Prof. von Hoff is trotting around like a teenager, knowing these sorts of discoveries. So,
oh yeah, I just remind people who, you know, are familiar with that. He was involved clinically in the development of Bisantrene back in the early 80s. So you know, he's very excited by this. Yeah. So anyway, so getting back to it, you know, I've actually got a document somewhere that's got, right, these are the experiments I want done to put into a business development deck. So it's very, very integrated. You know, we're really thinking about, right, how do we prove this out, you know, beyond any reasonable doubt? How do we explore the opportunities for combinations that are going to get a company who's got a drug that works by inhibiting that pathway interested in what we're doing? You know, so we're being very, very strategic. Yep, people want a deal. But as I said before, we want to deliver a really big deal. And, you know, that always takes a bit more time. And again, back to what I said earlier, if we'd done a deal last year without really working this all out and working out in the future to an extent where we can best deploy the drug, we would have left a massive amount of value on the table. We would be kicking ourselves, and I think probably shareholders would be out the front door with pitchforks and burning torches. So I think
the discovery was made.
I would also, you know, we shouldn't lose track of, we're all talking about this new exciting opportunity here. There is still a massive opportunity in our existing clinical program of cardioprotection anti-cancer. This is independent, I guess, in a sense, that we're talking purely anti-cancer in this particular case, use in purely anti-cancer. We have this whole entire program built around this cardioprotection anti-cancer program. None of that has gone away, and none of that's become any less valuable. In fact, it's become more valuable as we know more about the anti-cancer side, instead of being seen as necessarily a boring chemo adding with another boring chemo. This is saying, okay, we've got something here, starts to explain why we're getting this synergy between doxorubicin and Bisantrene. But it also goes to that value, the value of the cardioprotection. That's still just as huge, massive market alone in that. And that hasn't changed at all. This is just all on top of, this is why it's kind of crazy. We just keep adding more and more and more value on top. And the prize gets, you know, the potential prize gets larger and larger.
Yeah, and that aging effect more broadly. It's something we're going to tease out in the clinical studies. We'll you know, hopefully get a read on that. You know, again, you know, if we can convince people that it's going to protect them from the aging effects of doxorubicin, that could be huge. So, you know, looking at kidney function, epigenetic clocks, all sorts of things like that in the clinical data. So
and I think we shouldn't lose track of AML, like the original. This is what the drug was approved for. It's been all the way through. The opportunity, it's not in comparison to the opportunities this offers and the cardioprotection anti-cancer offers. There is still an opportunity there that most biotech companies would actually be extremely happy to have that. If they had a drug, at phase 3 stage, ready to go in for AML with the efficacy that, EE Bisantrene has, they would be extremely happy. We're just so unbelievably blessed with opportunities that we can go, oh, it's kind of minor, but it's actually a real potential opportunity there, particularly for fast route to market. So to bring a drug to market and get it onto the market has real value as you explore out all the different opportunities that are out there. So, there's still a lot of, in the historical data that we have, there's a lot of value still there. So none of it's gone to waste. Well, maybe some of it has, but the vast majority was spent on useful things. And we've been able to really generate a huge amount of value from a relatively small number of dollars.
Yeah, well done. Okay, we might just make this the final question because we're, over time in everyone's diaries. Daniel, for you, how significant is ESMO for the company? So you're going to be in Berlin for ESMO the week after next. Can we just talk about what that is and why it's important?
This is where we're releasing. So we're currently embargoed at the moment on the cardioprotection mechanism of action. And I'll be presenting a poster there on that. That'll be released to the market shortly afterwards. And so people will then get an understanding about the mechanism of action that lies behind cardioprotection. The big question there has always been, we've shown it in animals, in cells, everything else. Is this going to work in humans? Are we going to be able to show cardioprotection? And we think through knowing the mechanism of action makes it much more likely. I'm convinced that it will work in humans based on all the data I'm aware of. But unfortunately, drugs aren't approved on me being convinced. It'd be a lot easier if that was the case. But it all works towards, bit by bit, de-risking and people feeling more confident that this is actually going to work. And it's a major opportunity. Cardioprotection, anti-cancer, get that to work. That's, billions of dollars a year potential market there, just in that opportunity alone, before we talk about MIC or anything else as an anti-cancer drug.
Yeah.
Excellent. Okay. So people can keep their eyes out for that. We'll release that poster to market.
Yeah, it's coming up week after next. So yeah, so it's not very long. Not too many more sleeps for all of you shareholders that are anxious to know the mechanism of action around cardioprotection.
Very good.
Christmas is coming.
Great. So we've still got a whole lot of questions in the queue, including, I'll just mention a couple of questions that relate to people's personal situations, which we'll get back to you on those rather than sort of going into them here, but please expect an email from us. But with that, Pete, maybe I'll hand back to you for any closing comments.
Yeah, look, I think I probably bored everyone to tears talking about MIC, but as I said, it is, one of the things we said when we were thinking about the mechanism of action is it's a very Googleable target. So there's a lot of information out there. And in the presentation, I put a link to PubMed, which is, I spend quite a bit of time at my weekends in PubMed, as does the rest of the team. We have on Teams, we have a place called Papers, and every day somebody's posting something interesting in there. And it's, it's not just one person who's doing it. Across the company, people are finding interesting things and putting it there. So yeah, look, it's been an amazing journey so far. I had no expectation that we would be sitting here talking about this mechanism or anything like it. So it's a real turn up for the books. Again, full credit to the team of people behind it. That commitment and drive is what makes these things work. So long may it continue. So
excellent.
And thanks to everybody who've joined us. Thanks to all the shareholders who've supported this work. It's all part of a shared journey. We do enjoy sharing it. So, we look forward to what happens next.
Right, which includes seeing at least some of Race's shareholders in Hong Kong next week. So looking forward to seeing all those that can make it
Very much
along.
looking forward to that.
Excellent. Okay,
Thank
well,
you.
with that, thanks to
Thanks, Jane.
everyone from my side as well. And yes, pleasure. And I'll invite you all now to disconnect. Enjoy your night.